Good evening. Uh, this is another chapter uh, which is on growth of plant pathogens. And in this lecture, we will see uh, the characteristics of the plant pathogens with regard to their growth and development in the host tissues. And the basis of their characteristics, how the diseases appear or occur on the host plant, and uh, how the life cycle of a plant pathogen is related with the cycle of disease. I shall discuss all these things here. So before starting, uh, we must know about some of the terms which are basically uh, we need to understand uh, some of the terms related to disease development as well as plant pathogens. So, the meaning of parasitism or the meaning of pathogenesis and the meaning of parasite. In brief, what we can say that parasitism, which is a process which is caused by a parasite that may be uh, pathogenic in nature or may not be pathogenic nature, may be symbiotic in nature. As you can see, uh, parasitism of two types, where the one process is of the beneficial and another parasitism is of harmful in nature. So, how we can define the parasite and any organism which as a whole or in its part when depend on living tissues and which may or may not be pathogenic is known as parasite. And there are some, different categories of these parasites, and that also depend on how they multiply, how they survive, and how they attack or how they make their relationship with the host plant, like facultative saprophytes. And mostly the nature of these uh, microorganisms or parasites, basically they are parasitic in nature, but when the host is not available, they become saprophytes. Or sometimes we may also get another kind of parasite, which are basically saprophytic in nature, but as and when they get the host plant, they become parasite. Or we may also get another group of parasite which are truly dependent on living tissues and cannot survive without living tissues, and they are known as obligate parasite. And necrotrophs are another group where uh, pathogen, the first or the parasite, first they kill the host plants and thereafter they grow on those tissues. So, what should be the characteristics of a plant pathogen, uh, which can be classified as uh, a successful pathogen? So, a pathogen must have capacity to uh, grow inside the host tissues, colonize the host tissues, and can able to get nutrition from the host, or uh, have a capacity to uh, uh, break the immune system of the host plant, or also have a capacity to replicate. Pathogen also has a characteristic to exit from the host plant as and when uh, it completes its colonization, and do also have characteristics or capability to uh, disperse from the source of multiplication, and could be able to get the new host. All these are the characteristics should be there uh, in the organisms which are classified as a plant pathogens. And we also know about the nature of the pathogens, how they have their host range or their capability of attacking different hosts. Uh, Maybe they cause local infection, or maybe they cause the systemic infection. So the specificity of the pathogen uh, has various degrees. For example, some of the pathogens, they only target one species of the plant, while other pathogens, they attack only one genus of the plant. And there may be other pathogens which have a wide range of hosts belonging either to the same family or different families. So in this way, the pathogens, they have a different uh, kind of nature, and on that basis they differ among each other with respect to the point of infection or growing on the host tissues, maybe on the roots or stem or leaves or fruits or vegetables, or maybe on the tissues, maybe on the in the phloem tissues, maybe on the xylem tissues, or sometimes maybe in the cortical region. And pathogens may also have a nature to prefer uh, young or succulent tissues, or prefer older tissues. So in this way, pathogens have a different uh, kind of nature, and on that basis, uh, they cause the different forms of diseases. And when we talk about the disease, we must know about the inoculum potential, and the disease cycle. So inoculum, as we can say that the proper use of the plant pathogens which are infective, and when come in contact with the host, they have a capability to cause the infection. So those propagules which inside the infection are known as inoculum. And inoculum potential is the resultant of the action of the environment, and the bigger of the pathogen uh, which is to establish an infection. Then the success of the inoculum or the potential of the inoculum is also depend on the type of the host, or the susceptibility of the host and the amount of inoculum present on the host determines uh, the successful of the infection, or the parts or the host organs which are to be infected by that inoculum as also determine the successful of the infection or the development of disease. But the most important one is the inoculum potential, which determine the success of infection. Then before understanding the growth of the pathogen, we should also know the disease cycle, and the disease cycle we can define that disease cycle is a series of events uh, which lead to the uh, development of disease and growth, as well as the multiplication of the plant pathogen in the host. And that those events include the survival, the dispersal of the inoculum reaching to the host surface, the inoculation, and thereafter the activities which take place in the form of pre-penetration activities, then the penetration caused by pathogens, and the growth of the pathogens, reproduction of the pathogens. So all these events come under disease cycle, and it is also known as pathogenesis. And uh, it starts with the survival of the pathogen or perpetuation of the pathogen. Thereafter is spread, reaching to the host, then causing infection, then growth and development in the form of colonization of host. All these are the events, and which is also related closely with the life cycle of the pathogen. Then there is another term, pathogenicity. Pathogenicity can be defined as the is the ability of a plant pathogen or parasite to cause the abnormalities in the form of symptoms on host plants. If we can diagrammatic give the presentation of a complete pathogenesis or disease cycle, then we can start with the dispersal, then attachment to the host, then the acceptance or recognition of the host, then the germination or penetration by the pathogen, then causing infection, and after infection, the growth inside the host tissues, and as a result of the growth, there are the development of various abnormalities in the form of symptoms or like lesion development, and again the sporulation, and then after sporulation, again the pathogens either they reach to the perpetuation and thereafter the dispersal. So this is a kind of disease cycle, and you can see this is a diagrammatic cycle of apple escape, a disease which is caused by Venturi and Equalis. And here we can see the mode of survival through the pseudothesia, and thereafter the development of ascospores, and the primary infection which is caused by ascospores, and the continuous multiplication of the pathogen through production of conidia, where we can see here. Or this is a disease cycle of heat rust pathogens, where different stages are completed in uh, the life cycle of rust pathogen. And here also uh, the two types of the cycle we can see. The one is the repeated cycle, where the formation of iridospores on wheat and the continuous production of iridospores lead to development of the wheat disease. And later on, after the completion of the first this stage, the pathogens then it undergoes to the different stages, the formation of teletospores or teleospores, then thereafter the germination of teletospores after a resting stage, and then formation of basidium and basidiospores, and then again the basidiospores they cause infection on another host, which is an alternate host known as Barbary plant, where the spermogonial or pycnial stage and spermaceal stage as well as the acial stage will form on that. And again, when the acidospores produce, they cause the infection on wheat crop, uh, and the disease develop, and again the iridospores are formed. So in this way, the cycle is completed, and then there may be a different form of the events that take place in the process of disease development. And here we can see the inoculum, primary inoculum, secondary inoculum, then the density of inoculum, how the pathogen they causing infection, colonizing, then how they perpetuate, how they survive, and how they disseminate and form the host. So on that basis, there are two types of the cycle: basis of the inoculum or basis of the nature of the pathogen. The first one is the primary disease cycle, where there is only one cycle of the plant pathogen associated with this disease, and therefore these diseases are also known as monocyclic diseases, where the primary inoculum causes primary infection, and thereafter the overwintering of the inoculum take place, and then the dissemination occurs, and again they cause the infection when the new crop is planted in next season. So in case of monocyclic diseases, there is only one cycle of the one life cycle of the plant pathogen. Whereas in secondary disease cycle, also which basically uh, which cycle comes under polycyclic diseases, where the primary infection is brought by the primary inoculum, and thereafter secondary infection is brought by the secondary inoculum. And as a result of this, the production of secondary inoculum, reproduction and dissemination takes place, followed by overwintering and summary. In this way, you can understand the generic disease cycle, where uh, uh, the primary cycle uh, is basically uh, for the overwintering of the plant pathogen, and where the pathogen they undergo either through the sexual reproduction or sometimes asexual reproduction, and they survive without uh, the host, and then they cause the infection. Whereas the secondary cycles are repeated cycle, mostly it is being completed through the uh, asexual reproduction or asexual cycle, you can say, and where the pathogens reproduce asexually, forming different forms of the spores, which are disseminated, reaching to the host surface, causing infection. Then the incubation period is there, and then disease development is taken place. So in this way, the cycles, these cycles, secondary cycles, they continue till the crop is there. So there are several cycles. So how we can say which are the primary inoculum or which are the secondary inoculum? So the primary inoculum, by which the pathogens they survive uh, in the absence of host. So the primary inoculum may be oospore, or maybe the dormant hyphae or mycelium of the plant pathogens, or maybe the sclerosia or hard structure of the pathogen, maybe the cyst or the galls or the cockles of the pathogen. Or in case of nematodes, the cockles or galls are there, and all these are the not only the oospore, maybe the ascospores, which also act as a primary source of inoculum. Then come to the secondary inoculum, where uh, the conidia, which produce uh, in a large number and repeatedly, or maybe iridospores in case of the rust fungi, maybe zoospores in case of the fungal pathogens belonging to phycomycetes, or where the zoospores are formed inside the sporogen. So all these are the secondary inoculum. Then what are the sources where the inoculum survive, and after the survival, the inoculum causes the infection. So most of the plant pathogens of which the inoculum survives in the soil, maybe the bacterial pathogens or maybe the fungal pathogens, uh, the inoculum of which in the form of different propagules survive in the soil or on the infected plant parts, maybe on the seed or maybe on the cuttings or maybe on the propagating plant material or maybe on the infected uh, uh, or diseased infected plant debris or disease debris. So there are pathogens in case of Phytophthora, the Phytophthora pathogen, it survives on infected plant debris. Or written area, it survives on infected plant debris. Or maybe some of the uh, other host, maybe other alternate host or collateral host, where the pathogens survive in the absence of host, like in case of rust pathogens. The rust pathogens, they survive on alternate host. Or in case of the viruses or mycoplasma like Aeroporism or phytoplasma or fastidious bacteria, all these survive on the collateral host. Some of the fungal pathogens like powdery mildew, fungi, they also survive on collateral host. Then how the inoculum is being carried out by different agencies. Later on, we will discuss when we study, when we will discuss in the chapter survival and dispersal of the plant pathogen. But basically, we should know that how the inoculum is being carried out by different agencies. Like in case of powdery mildew and downy mildew, rust, all the inoculum of all these diseases is being carried out by the wind. Or bacterial pathogens, mostly they disseminated by the water, or some of the fungal pathogens also disseminated by the water, or the soil-borne plant pathogens, maybe fungal pathogens or bacterial pathogens like Xanthomonas or Sclerosium or Rhizoctonia or Sclerotinia. All these of the pathogens and the inoculum of all these pathogens is being carried out by the soil or the viruses or the phytoplasma, maybe fastidious bacteria. The inoculum of all these are being carried out by the insect vectors, uh, maybe hoppers or maybe silent insects or maybe white flies or maybe uh, uh, some of the uh, other sucking pests. Our inoculum may also be carried out by the uh, uh, infected tools uh, or planting materials like that. And some of the plant pathogens, they do have a capability to, to move from one place to another place. And they find the host without having any involvement of any agency. So like the larvae, juvenile larvae of Phytophagus nematode uh, uh, move uh, in the presence of thin film of water and find the host and cause the infection. Likewise, the juice spores of fungal pathogens, they also uh, move from one place to another place and cause the infection. So the inoculum has two different types of sources, the inside and outside sources. Inside source means when the inoculum is present on the plant or plant debris or on the soil as a result of the uh, attack of plant pathogens and as a result of the development of disease. And uh, the inoculum of these plant pathogens, which is produced on infected plant parts, maybe on the leaves or maybe on the stem or maybe on the trunks. So all these are the inoculum known as inside sources. Then another term is the inoculum, which comes from the outside sources, and where the inoculum, when it comes from another field, when it comes through the seed or through the transplanting material or through the propagating plant material, so all these inoculum, uh, when they introduce in the new area, these inoculum are categorized as the inoculum from outside of the source. Then uh, how the inoculum uh, reaches on the host. So in some cases, the inoculum which is produced on the plant surface, like in case of fungal pathogens or bacterial pathogens, maybe or nematodes or parasitic plants. So the inoculum is mostly produced on the infected plants, or their inoculum reaches to the plant surface when the infected tissues break down. However, the inoculum uh, produced by the viruses, virides, molecules, fastidious bacteria, and protozoa within the plants. And all these inoculum may be carried out by the wind, water, or insects. Then what happens in the process of inoculation? Inoculation is the pathogen or any part of the pathogen that contacts with the plant at certain site to initiate the infection process. So in that case, the spores or the fragments of the mycelium or sclerosia, they come in contact with the certain sites of the host plants uh, in the form of inoculum, and uh, thereafter they starts uh, developing to cause the infection. Or in other cases, uh, like in bacteria, molecules, protozoa, virus, and virids, uh, that uh, there are no spores. So the inoculum is present in the form of intact cells. Then, as we discussed earlier, that the primary and secondary inoculum, uh, uh, which is uh, either uh, give the uh, survival of the plant pathogens or which give the further infection and multiplication by the pathogen. Then how the pathogens they starts penetrating the host tissues, and penetration can be defined as is the process by which the pathogen enters its host. That the process of penetration it varies among the different plant pathogens, and the complete the whole penetration process may be divided into three categories. The first one is the pre-penetration, the another one is the penetration, and the third one is post-penetration process. So in case of pre-penetration, the pathogens or the propagules of the pathogens started uh, developing in the form of infective propagules. And as and when uh, the pre-penetration process is completed, the pathogens start penetrating the host tissues. And after the penetration or after the entering inside the host tissues, the further growth and development comes under the post-penetration process. So there may be the direct penetration or indirect penetration. So some of the fungal pathogens as well as the nematodes, they have capability to uh, break the barriers and they penetrate the host tissues. But uh, some of the fungal pathogens or mostly the bacterial pathogens or viruses or viroids or phytoplasma. So all these are the pathogens. They do not have a capacity to penetrate host tissue directly. Therefore, uh, they enter either through the natural opening or through the wounds caused by mechanical injuries or some the primary invaders, uh, or uh, maybe by the uh, injuries caused by insect vectors. Then, what is the process which takes place under the pre-penetration activities? So, the first one is that the propagules of the pathogen they make the attachment to the host tissues, or they make the attachment to the host, rather to say, or different parts of the host, maybe on the root, or maybe on the leaf, or maybe on the stem. Then the second process is the germination of spores. If the spores are attached to the host, then the further the spores they germinate, uh, if, especially in case of the fungal pathogens, the spores germinate in the presence of water, and in case of bacterial pathogens, the bacterial cells first they multiply uh, on the surface where they attach to the host surface before entering inside the host tissues. Or in case of the nematodes, the egg hatching process comes under pre-penetration process, where the release of second star larvae takes place. So all these are the pre-penetration activities. Uh, then uh, when we say about the pre-penetration activities, so the pre-penetration activities do not occur in case of those pathogens which are not having a capability to enter themselves inside the host tissues, like viruses. Viruses always get entry either through the mechanical transmission or through the injuries inside the host plants or by the vectors. Similarly, bacteria, they also enter through the or by the natural opening, like stomata or lenticels are either two, or the wounds or injuries caused by uh, some of the mechanical means or sometimes by the primary invaders. Whereas nematodes, uh, they do not need any kind of uh, uh, natural opening, they get entry through the direct penetration. Then, how the uh, attachment of the pathogen takes place on the host. So, what is the process which is uh, develops uh, in the form of pre-penetration activities? So, here uh, then some of the pathogens which directly penetrate to the plant tissues uh, and uh, by the vectors, and then they're surrounded by the cytoplasm, such as molecules, fastidious bacteria, or viruses. In another case, the pathogens, they externally they come in the contact with the host plants, and then they penetrate the host tissues, such as fungi, bacteria, and parasitic air plants. So the adhesion of the pathogen with the plant surface is carried out by the secretion of the some mucilaginous substances uh, uh, on the host surface. And these are the substances or compounds which are made up of either insoluble polysaccharides, or glycoproteins, or lipids, or some fibrillary materials. And they moistens and they form a sticky uh, area and which helps to attach the propagules of the pathogens. Like in some of the fungi, like powdery mildew fungi, some of the enzyme, like cutaneous enzyme, is being released and which makes the plant and spores areas for the attachment on the host surface. Then there is also uh, a determination of uh, or the importance of the quantity of the inoculum. Some of the pathogens, they do require a higher quantity of the inoculum for the development of disease, whereas some of the pathogens, they require less quantity of inoculum. In case of